kids, welcome to this interesting lesson in which now we take a look at the next important type of mutation, that is we talk about the gene mutations. So, are we trying to talk about mutations in the complete gene, which is a segment of DNA? Yes. For example, let's say this is my gene, that is a functional segment of DNA. This gene will be made up of nucleotides. We know what nucleotides are, right? We have seen this in the lower class. What exactly is the DNA and RNA structure? So, these nucleotides are always present in a series. When I say one particular gene for one particular protein, that means it is having a series of amino acids. That means it is going to definitely have a series of codons, that is all the codons which are present in one particular sequence. What if this sequence changes for some reason? That exactly would define my genetic or gene mutations. Thus, I would define gene mutations as abrupt, sudden or what you say unexpected changes in the complete segment of gene. And what kind of changes are we expecting here to happen? We can expect the complete nucleotide sequence. We can expect the small portion or series of codons being lost, series of codon being added, series of codon being substituted also. Yes, for example, A is substituted by G or something like that at one point, or many codons together. All these kinds of abrupt random changes happening in the genes is what we are studying under gene mutations. So, now let us take a simple example for this. Let us assume I take just one gene. Yes, I am taking one strand of DNA, I am not taking the other. We know it is a double helical structure, but we just take an example of say A, T, G, C, G, C, T, T, A, G. Yes. For example, this is one sequence. Yes, I am not just pointing out the codons which are present. Let's assume this is one sequence continuing together. Now, in this, what are the possibilities that can happen? What kind of changes do you think can happen in this? One changes, some part gets deleted. One changes, some part gets duplicated. One change is where some part is substituted by something else. Correct? All these possibilities gets us to the types of gene mutations. So, I would say the first type of gene mutation would be substitution. Substitution of what? What are we substituting? Can I substitute this G by either C or T or A? Yes, I can. I mean some kind of changes happening so that this G changes to something else. Now, here let us assume this is changing to another A, this G getting changed to A, right? That means my purine is getting changed to another purine. Yes. What if this gets changed to a C? That means my purine gets changed to a pyrimidine, correct? So, purines changing to purines will be one type, purines changing to pyrimidines and vice versa. Pyrimidines changing to pyrimidines or pyrimidines changing to purines will be another type. Correct? So, two major types of substitution that we get, which is transition and transversion. Yes, let us talk about the first one, that is transition, and then talk about transversion. Transition will be where my purine is replaced with another purine or pyrimidine changing to another pyrimidine. Yes, so same changes the same. Yes, so my purines getting changed with purines or pyrimidines changing to pyrimidines. Yes, that would be transition. Transversion will be exactly opposite. Purines changing to pyrimidines and vice versa. Correct? that would be my substitution. Yes, this is where one or maybe two or more genes, nitrogen bases are getting changed. That is substitution. What if, say here itself, yes, what if the next possibility of inversion of a sequence takes place? For example, let us assume this portion, yes, this gets flipped. The sequence will turn out to be T, C and G. That means the complete sequence has now got changed, correct? So, I can have inversion at one or many places, yes, inversion which is taking place. Now, similar to inversion, I can have, because of inversion also, there are changes in the sequence of amino acids. Now, because amino acids are changing, the protein produced will be different. That is the reason why sequence of amino acids will give rise to different type of protein. Hence, inversion is also important. So, next one will be my